Okay, chapter 3 is on cells and also membrane transport, enzyme function. Let's jump into it. So we're going to talk about cell theory. This is the idea that everything's made out of cells. We'll talk about surface to volume ratios. Basically, the smaller the cell, the more surface it has for a given amount of inside, and that makes it easier to do gas exchange and exchange things across the cell's plasma membrane. That's the outer membrane. And yes, in this class, I want you to call it a plasma membrane. And we'll talk about microscopes and why they're important. Anyway, moving on. So a cell is the basic unit of life. Everything that we know of that's alive is made up of cells. Now we have some weird things like plasmodial slime molds that switch back and forth between being single-celled and multicellular. Very strange stuff, but they still all have cells. And new cells only come from other cells. That is obviously something that is a new finding that we didn't have back in the day. Pasteur and people like that who helped pioneer things with microbes, they explained all that. Anyway, moving on. So here we see a blood vessel packed full of red blood cells. Here we see nerve cells. There we see osteocytes. Osteocytes. Those are bone cells living in lacunae, little lakes. They're connected by canaliculi, little canals. Anyway, those are mature bone cells, and they detect stress in bones and warn other types of bone cells, osteoblasts that build bone matrix, and osteoclasts that cut it apart. Sorry, that was my hand trying to do scissors for cut it apart. That's my mnemonic device to teach the kids osteoclast cut bone. Anyway, let's move on. So cells are smaller because of their surface to volume ratio. If a little cell has a lot more surface for the given amount of inside, and that makes it easier to pass nutrients. And that also makes a limit to how big a cell can grow. So here they have an example. They're looking at a 4-centimeter cube, okay? And if you break it into smaller cubes, you got the same amount of stuff inside, but you have a lot more surface area, and that makes it easier to pass things across it, across the sides, across the membrane. So when you look at the ratio, surface area to volume, and your one big cube, it's 1.5 to 1. That's not real good. You break into a bunch of tiny little cubes, you have a 6 to 1 ratio. Way better. Much easier to exchange nutrients, exchange gases. All right, let's move on. So microscopes, there are different types of microscopes. I've actually got a video that I put together recently about microscopes. And let me pull it up real quick, and I'll put a link to it down below for y'all. Okay, I just pulled up 2 videos that I will put the links to down below so you can look at them. Videos I've made about microscopes. One is the intro video that I use for microscope labs, but the other one is actually really, really cool. It talks about microscopes that you're not going to see in most places. Things like atomic force microscopes, which are amazing. They feel the surface of a molecule to tell you its shape. And electron microscopes, different types of electron microscopes. Cool video, especially because it's, you know, hands-on. Here we are, we're looking at and using these microscopes, seeing them firsthand. It was amazing. That's photos from a trip that I was able to take with some students to the University of the Antilles, and way cool stuff. Anyway, moving on. So microscopes have different types of different types of sources of energy going through them. So the compound light microscope, you know that it has light for the beam of energy, and light microscopes have glass lenses. Uh, let's compare that to, say, an electron microscope. Instead of a light microscope, an electron microscope has a beam of, pretend to be shocked, electrons. And because electrons have a negative charge, we can manipulate them and move them with magnetic fields. So instead of the lens being glass, the lenses are donut-shaped electromagnets, and you shoot the beam through the hole, and then you turn up or down the current on that magnet, and that will control the width of the beam at that spot. So in a light microscope, glass lenses, you move the distance between the lenses. You get the same effect of narrowing the beam, but it's by moving this lens away or moving this lens closer. Electron microscope, you do it with magnetic force through electromagnets. Anyway, moving on. Here we go. So we're going to talk about the typical light microscope and the transmission electron microscope. You can see the transmission electron microscope can see things that are a lot tinier. Why? Because a light microscope is limited to things about the wavelength of light. If you have two things that are closer and you're looking with a light microscope, you can't tell if it's two little things or one big thing, because it's just a kind of a blur at that point. You can't tell that far down, that highly magnified. A transmission electron microscope, because it's not using light, any electron microscope, because it's not using light, it's using a beam of electrons. Electrons, just like light, have a wavelength, but it's much smaller. So you can tell the difference between two things that are really close together. Anyway, moving on. So here we have a light microscope photo of red blood cells in the wall of a blood vessel. And here we have red blood cells in the wall of a capillary, way zoomed in closer. That's a TEM, a transmission electron microscope. Now, a transmission electron microscope is shining a beam of electrons through. It's transmitting the beam through the specimen, okay? You have light microscopes like that too. That's what they're showing there. The light microscope shines a beam of light through the specimen, but that's the compound light microscope. There's also what they call a dissecting microscope. It's a light microscope that when you focus and everything, it shines a beam of light on the specimen and it bounces off to the lens, and you can then focus. It's basically a real low power. It's like a magnifying glass almost, but it lets you look at the specimen. This is what they're talking about when they said lower magnification than other types of microscopes. A transmission electron microscope is like a transmission electron microscope is like a light microscope that shines a beam through it, transmits it through. A scanning electron microscope is like a dissecting microscope where you bounce the beam off the specimen. And you can see the difference here. Both of these are looking at red blood cells in a blood vessel, but we'll move on. All right, so a TEM, a transmission electron microscope, gives you a 2D image. You're looking through a slice. It uses a beam of electrons. You already heard that. Human eye can't see the image. That's because our eyes detect light, not electrons. And the image is projected onto a screen. One of those two videos, I have some nice photos of a TEM and an SEM. The TEM is the much bigger, taller one. Now, where it says high magnification, no live specimens, that's out of date. in reality, no specimens that will live very long, but you can run them in or at least not do well very long. But the reason why they're saying that is because the transmission electron microscope beam of electrons coming down, if there are any air molecules inside, it used to be that if a beam of electrons hits an air molecule, they get scattered all over inside the chamber and there's no way to figure out what's going on. But things that are happening in regular photography are influencing microscope photography. And the microscope, and just like we now have cameras that will do autofocus, we have electron microscopes that if things start scattering, like, oh, no, that's not right, ignore those scattered electrons, just focus on the ones that are doing what we thought they should do, that are passing all the way down. And you can remove some of the noise automatically and the image is better. What that means is you don't have to pump all the air out of the chamber. So we do have some electron microscopes now that the way they work is above, they have many chambers. Like when I used an electron microscope, we had 2 different pumps to pump out all the air. When I went to visit University of the Antilles, they had microscopes that they pumped all the air out of this chamber, and then this chamber, the next one down the column, and yeah, they're connected. There's a hole that the electron beam comes through, but it's a really tiny hole. This one is most of the air is pumped out. And the one below that, some of the air is pumped out. And the one below that, almost none of the air is pumped out. By having lots of vacuum pumps at all up and down the height of the column, they could keep some air in, and if they're looking at a bug or something, they don't have to kill it to do the thing, to look at it. So where your book is saying high magnification, no live specimens, it would be better, more accurate, say high magnification, uncommon to use live specimens now. Used to be impossible. Now some microscopes can. Anyway, let's move on. All right, a scanning electron microscope. I love these. These are so much more fun to work with than TEMs. This produces a 3D looking image. This is like a photograph. This is a lot more like, when you work with a TEM, it's a lot more like a microscope photography class. Anyway, or photography contest. You got a beam of electrons, they're bouncing off the specimen, and again, high magnification, no live specimens. I think that's probably true here. But the part that's really cool, the SEM, they now have attachments that can chemically analyze the specimen, partially. So if you're shining a beam of electrons on something and they bounce off, they could knock some of the molecule apart, break some of the surface molecules, knock atoms off it and stuff. And we have detectors for that now. So it's a microscope, and it may not be able to say, oh, that's copper sulfate, but it would be able to say, hey, you know, there's a lot of copper on this thing in that area that you're looking at. And so it's a microscope. When you're using it for material science, it can literally tell you the chemical composition roughly of the thing you're looking at. And that's really interesting and really useful. All right, here's a check your progress. You know the drill. I'm going to pause for a second and then we'll carry on. You can, at this point, answer the questions in your head. You move on. If you can't, you roll back to the start of the video and watch it again. All right, so now we're going to talk about cells, the difference between prokaryotes. Those are things without a nucleus. I'm talking about things like bacteria, archaea, and eukaryotes. Those are things with a nucleus. The name literally means eu is true or with, and karyo is kernel of corn. It's a Greek word, which doesn't really have anything to do with corn. It's just we wanted a word for something really tiny and really important. So nucleus got stuck with the Greek word for a kernel of corn. Anyway, so eukaryotes are anything with a nucleus. I'm talking about animals, plants, mushrooms, fungus, and then this weird grab bag that we shove everything into that we call the protistas. And we'll talk about what plasma membranes do and the organelles in a cell, the mini organs. Hold on a second and let me pause this. Okay, so we're going to talk about cells, talk about prokaryotic cells, eukaryotic cells. What's the difference? Basically, eukaryotic cells are a lot bigger, have a lot more parts. The most significant of those would be the nucleus, okay? Eukaryote means with nucleus, actually means with kernel. We co-opted a Greek word, karyo, to mean, which really means kernel of corn, to mean nucleus. Anyway, so prokaryote means before nucleus, and eukaryote means with nucleus. Prokaryotes are things like bacteria, or very similar to bacteria. Those would be the archaea. They're very small. They're about the size of a mitochondria. We'll talk about reasons why later on. Anyway, but they're a much smaller cell, and they have fewer parts inside. They don't have all these membrane-bound organelles. But let's move on. The plasma membrane, I want you to call it a plasma membrane in this class. The plasma membrane is the membrane around the outside of the cell, all right? That membrane around the outside of the cell, don't call it a cell membrane, call it a plasma membrane. The reason why is you then are it then becomes easy to remember things like, oh, well, the stuff inside of that is the cytoplasm, or oh, breaking that outer membrane is lysing it, breaking it, is called plasmolysis. So if you call it the plasma membrane, it's easy to remember the other stuff. And so let's move on. So prokaryotes don't have a nucleus. I'm talking about the bacteria, the true bacteria, eubacteria. Remember, eu means true or with. And the archaebacteria, the ancient ones. Okay. Their DNA is a little different than the other bacteria. Sorry, their DNA is different from the true bacteria. Now, eukaryotes, things that have a nucleus, that's animals, plants, and fungi. Those are all going to be real familiar to you. The protists might be weird. Protista is a big grab bag that they threw a bunch of stuff in. Things that it's probably, I'm going to use a science word, it's probably not monophyletic. Meaning these things are probably not all that related to each other. They just lumped them together. What I mean is, like, algae are going to be in Protista. But algae sure seem a heck of a lot more like a plant than they do like an amoeba, which is also in Protista. Anyway, so let's move on. Here you see a cell, and this is a eukaryotic cell. You know that because you've got a nucleus, and that dark spot in the middle, the nucleolus, right there you are reading the DNA inside the cell. The DNA stays inside the nucleus most of the time, except when you're doing cell division. But yeah, DNA in the nucleus, and this nucleolus is a spot where you're actually reading the DNA and copying that information into RNA. But the nucleus, so eukaryotic cell, but there are other things too. All of this stuff, the endoplasmic reticulum, they divide that into smooth and rough. The rough is rough because it's covered with ribosomes. And the endoplasmic reticulum with ribosomes on it, we'll talk about later. Ribosomes like little chefs that cook up proteins. This is a protein making site. It's got lots of little protein makers on it. So I think of it as a factory full of chefs cooking up meals. Anyway, so let's move on. Oh, the other thing they have labeled here is chromatin. Chromatin is DNA wrapped around histone proteins. So DNA is not just out there as DNA. The DNA is packaged. Think of thread. When you get thread, it's wrapped around a spool. That's how it's packaged. Makes it easier to handle and work with. Same deal. DNA is wrapped around histone proteins. And the combination of DNA wrapped around histone proteins is what we call chromatin. All right, so let's move on. Here we have a drawing. It shows more parts. You can see sometimes there's so much endoplasmic reticulum, it's hard to see other things. So here you can see other stuff. This is figure 3B. We're going to go over all these things, but this is a good high-level overview of all the stuff that you need to know. I'm looking right here. What are microtubules, intermediate filaments? Where they're saying actin filaments, a lot of people call those microfilaments. Actin is the type of protein. They tend to be made out of those, so yeah, that's fine. Anyway, yeah, this is a spectacular slide because it has all three parts of the cytoskeleton. It has what centrioles do, and yeah, they're made out of those microtubules that are part of the cytoskeleton. That's great. Those are the microtubule organizing centers that when the cell does cell division, it's going to grow what they call the spindle, the mitotic spindle. And the chromosomes will be pulled apart by those, the shortening of those microtubules. Anyway, the centrioles where they're going to grow from. Before the cells are ready to divide, the two are like this. When the cell's getting ready to divide, they're going to migrate to the poles, and then they're going to grow spindle fibers, and they're going to pull on chromosomes. Anyway, that's what that's for. So let's see, lysosomes, yeah, to break things. Yeah, I would say every single thing on figure 3.4b is something that is valid to test on, something you ought to know. That great for review, great for studying. Know this illustration. Moving on. Okay. Whether you're talking about a prokaryote or eukaryote, a prokaryote being a simple cell, eukaryote being a complex with like lots of parts like we just saw, they got a membrane around the outside. We call it a plasma membrane. That membrane surrounds the cell. It's made out of a phospholipid bilayer. You've heard this before. You know, phospholipids are lipids that are special because one end of them loves water and one end of them hates water. So all the heads will line up towards the water on the inside. Another layer, all the heads will line up towards the water on the outside. The tails, which hate water, point towards the middle of the membrane. So that means here, water, middle, no water. And things that bring lots of water, have lots of polar charges, they don't cross that membrane very well. Anyway, so that's what the selectively permeable is. It controls what gets across and what doesn't. Things that are big, that have charges, they don't cross the membrane well. Things that are small, things that don't have a charge, they're much better. All right, inside of that plasma membrane, we already heard this, you've got cytoplasm. Cytoplasm is actually made of cytosol plus organelles. The thick liquid inside the cell is cytosol. And organelles, the name sounds, the L is diminutive. What I mean by that is, you know, in Spanish, if you want to talk about something being small, or the little version, you put ita on the end. In German, you would do the same thing, but you would put klein. In Japanese, you would put ko. In the same way, organelle is a mini organ, all right? A little organ. These are things inside the cell that have little jobs to do, just like the organs in your body have little jobs to do. I'm going to give you the most extreme example. If we look right here, where was it? Lysosome, okay? A vesicle containing digestive enzymes. Yeah, that's nice. Let's go one past it. It has digestive enzymes. It has acid. This is a little tiny mobile floating stomach, basically. That's the stomach of the cell. The cell brings in something and then decides, oh, I need to break this down and digest it. It fuses a lysosome with it, which spills out the enzymes and the acid, and the thing gets digested. Now, the lysosomes are not just used for that. A cell can also break down, digest, and reuse, recycle the parts of its own organs, its own little parts when they're starting to wear out. But anyway, a lysosome is an organelle, like a little organ. It's just like a little stomach. It has digestive enzymes, it has acid. That's what it does. All right. And all those things that we were talking about there. We were talking about the centrosome and the lysosome and the mitochondria. Those are all organelles. Okay. Membrane-bound organelles. Some people argue about whether or not they want to call a ribosome an organelle. It's really sort of a moot point because the ribosome we know is the protein maker, the little chef that cooks up proteins, and the roughy R is covered in them. So that organelle, yeah, whether you want to say that the ribosome is an organelle or not, there's certainly an organelle covered in ribosomes. It's roughy R. All right, so early cells were prokaryotes, the archaea, the ancient ones. When the atmosphere has no oxygen, it's things that are there, they don't survive well in oxygen usually. Things, the book is talking about the opposite. It's saying, oh, it'll be hard for things that are around now to survive there. Yeah, it's true. But something that's probably more interesting than this, hyperbaric medicine. There are microbes that are what we call obligate anaerobes. They can't survive in oxygen. And one way of treating, and unfortunately, some of these are things that are antibiotic resistant too. So you can't treat them with antibiotics. What do? You take a patient and you put them in a hyperbaric chamber so you can give them extra oxygen. And if you do that a couple hours a day, you can eventually make the human body inhospitable to microbes that can't handle oxygen. So it's pretty cool. It's a good way to get rid of them. Anyway, moving on. Back to what they're saying here. Early atmosphere is thought to not have had oxygen. The main reason for that is because oxygen tends to be very corrosive and break chemicals down. And when it's saying some archaeans still thrive in very inhospitable conditions, hot vents, totally true. But we now know of other places where they survive. The gills of shallow water clams, for instance, all kinds of crazy stuff. We'll talk about that more later. But right now, this is what you need to focus on because we're doing human biology. We're not doing an, oh wow, look at the amazing weirdness of life. So let's move on. Eukaryotic cells, we think, probably came from archaea. The reason why is the organelles that are inside. You see here this word endosymbiosis. Let's break that down. Symbiosis is same life. Same, bio, life. So things are living together. Endo means inside. So it's things living together because something was taken in and put inside. Now, before we continue with this, let me pause a minute. Okay, folks. I want to show you some things before we go on with endosymbiosis, because if we don't, you're like, whoa, that's just crazy. But I want you to make it clear we see things similar to this going on all the time. So here we have lichen growing on a tree. This is like an encrusting lichen. Here we have like a foliose, which means, oh, it looks kind of like a plant. But what I want you to know is, oh yeah, totally. A Nat Geo video. I'll give you guys that video in the links below. But if we look right here at lichen, what we're actually dealing with, oh, and in Florida, you have like deer moss sitting on the ground if you're in scrub areas. So lots of different forms of lichen. But what they are is algae growing inside of fungus, okay? So the fungus provides the house, it makes it moist, and that gives the algae a place to grow. The algae grows, captures light, does photosynthesis, makes sugar that feeds the host it lives inside of. They both survive that way because they're symbiosis, same life. Now, not all symbiosis is mutualistic. You know, a parasite living on a host, that's symbiosis. But in this case, it's the way that most people use the word symbiosis. It's mutualism. It's good for both species, all right? Now, so we've established that you could have, say, an algae living inside of something, photosynthesizing, feeding it, and they're both good. Let's go to over here. Here we've got coral, all right? And a lot of people don't know this, but the skeleton of a coral will have color. The coral itself, the little polyp, the little animal, it may not have any color. You can see this one's clear. What's colored are the zooxanthellae. Think algae again, living inside of this, okay? And as long as they are doing a good job of taking care of it, it's just like the lichen. The zooxanthellae capture light, do photosynthesis, make energy, and share the energy-rich molecules they're making with the hosts that they're living in. And as long as they're doing that, the host keeps them around. If they don't do it, this is what happens with coral bleaching. If the water gets too warm, then the zooxanthellae can't do their job well, and they actually start releasing things that are free radicals that can harm the coral. So the coral spits them out. Coral gets rid of the zooxanthellae, and it's not good in the long run because the coral gets most of its energy from the symbionts that live inside of it. So this can kill the coral, and that's what happens in coral bleaching. Well, if the bleaching lasts too long. Anyway, so you understand that. You understand that the coral will have a symbiont living inside of them until it's harming them instead of helping them, and then they get rid of it. Now, because the free radicals are given off. Now, there's another example similar to this. I mentioned it in passing. In some shallow water environments, we have mollusks like clams, like shipworms, that have microbes living inside of them doing chemosynthesis instead of photosynthesis, chemosynthesis. But the idea is that you still have a symbiont microbe. It's still living inside of the organism. It's still producing energy and feeds the host. The difference is that with, say, the little clams, if the symbiont stops doing the job well, they'll just digest the microbes. And then as they filter feed, if they find some other microbes, they'll re-inoculate themselves. But yeah, it's like, oh, you're not helping me? I'll just eat you. Anyway, so keeping that in mind, now we're going to go back to talking about endosymbiosis. You can see we're doing the symbiosis thing here. We're talking about things that are alive, that share a life together, and that one is inside the other. So we didn't really go too far off. What I'm going to do now is jump to another PowerPoint, one that I use with my other classes, and you'll see how it's related. Basically, it has a little bit, it's almost exactly what you're about to see in your book's PowerPoint, but I think that it has a teeny bit more detail in an important way. So hold on. Okay, so here's the endosymbiosis slide that I tend to use. Endosymbiosis, the idea is pretty simple. You have two simple cells. Wow, that was the idea is pretty easy to understand. You have two simple cells, a little one and a big one. And the big one takes the little one inside. And then instead of digesting it, it keeps it. So the idea would be, for example, a big cell comes in, grabs a little cell, pulls it inside. It's like, I'm going to eat you. Oh, wait, you'll make food for me? You make energy-rich molecules for me? Yeah, you can stay. I'm not going to digest you. Keep the lysosome over here. You just stay right there and do your job, and that's cool. And you reproduce inside of me, and I reproduce, and we're all good. Now, you might think that sounds fantastical, or whoa, how could that possibly be? Well, I already gave you 3 examples of similar things happening right now. But let's go beyond that, okay? Endosymbiosis, you already know, endo means inside of, within, sim is same, and bio is life. So the name literally means things living together, one inside the other. But when scientists say it, they're talking about how we got complex cells from primitive cells. And the answer is, we think they took in other cells, and then instead of digesting them, they kept them because these cells are making energy for them. So let's go right here. We have this idea, and a lot of people are, well, how could you possibly know that? There were no people there. See that? So I want to talk about the evidence. All right. When you look at the and the book here, sorry, the other book here is doing mitochondria and chloroplasts. But this is true for all the plastids. What I mean is the amyloplasts and the chromoplasts and stuff like that. There are a lot of organelles that have their own DNA, their own ribosomes, make their own proteins, and reproduce on their own schedule inside of the large cell. These organelles are doing their own thing, almost like they were their own little mini cell. And basically what we're saying is that scientists think they were once their own little cell. It's not just that mitochondria have their own DNA, it's that the mitochondrial DNA is more similar to the DNA of a bacteria than it is to the DNA of the rest of the cell. Like, we have a cell, we have a nucleus, we talked about that. The DNA in the mitochondria is more like the DNA of this bacteria over here that is like the DNA in the nucleus. Now you remember chromatin. DNA is stored as DNA wrapped around histone proteins. The shape of the histone proteins in the mitochondria is more like the histone proteins of a bacteria than the histone proteins that are found in the nucleus. Okay. The ribosomes, the shape of the ribosomes, little protein factories, the shape here is not quite the same as the ones in the rest of the cell. It's more like the shape of the ribosomes in the bacteria. So, and this is true for whatever species we look at. The things happening in the things happening in the organelles, the plastids, the mitochondria, they tend to look very bacterial in nature. And most of these things have a double membrane around them, almost like they had their own plasma membrane, and then when they were engulfed, when they were brought into the bigger cell, another membrane was around them. That's what you're seeing, double membrane in the mitochondria, double membrane in the chloroplast. So this is the type of stuff that we consider to be evidence. Let's move on. Whether you agree with it or not, you need to at least understand the concept for this class. So endosymbiotic theory, remember when we say theory, we think there's a lot of evidence. The evidence is that we keep seeing this everywhere we look. But endosymbiotic theory is that free-living simple cells took in other free-living simple cells a long time ago, and that's how we got organelles and became more complex. That's how our cells got more parts, basically. Those parts used to be other free-living cells. And I know the temptation is, oh, that's so nah. But if you think about it, it's not any different than what we're seeing happen right now with coral, with fungi. We have this happening right now. Anyway, so I'm going to go back to your book PowerPoint. Give me a second here. All right, so okay, so endosymbiosis, the idea that a big simple cell took a little simple cell inside, and then instead of digesting it, it decided to keep it there. Got it. And here is the here's your textbook's pretty diagram. It's similar to the simplistic one that I showed you before, but I don't think it's any better because it doesn't list any of the evidence, any of the modern current evidence that we have. So whatever, 6 of one, half does the other. You need to understand the concept of endosymbiosis, even if you have no interest in it, even if you don't think the idea has merit. Understand that the DNA, the ribosomes, the histone proteins do have evidence. And in fact, there's an antibiotic, I should have thought of this, that it is used against bacteria, and it works on bacterial ribosomes, but as a side effect, it can damage mitochondrial ribosomes. So again, the mitochondrial ribosomes more similar to bacteria than to the other ribosomes in the cell. Let's move on. Oh, it's check your progress. Okay, I'm going to pause for a little bit, and you can see if you can answer the questions. If not, stop the video, rewind it, and rewatch this section. Okay, let's move on. So we're going to do membrane transport. We're going to talk about how things get across. We're going to talk about plasma membranes, what they're like, how things get across them, diffusion, osmosis, facilitated transport. I'll tell you right now in advance, the diffusion, things are spreading out from an area where they're highly concentrated, where they're less concentrated. Osmosis, the O should remind you it is the movement of H2O. Osmosis is the movement of water. And both of those have the same goal. They're trying to reach an equilibrium where you have the same amount of stuff. So say it's very concentrated on this side of a membrane and it's almost pure water on this side of the membrane. If you try to even things out by having water go across the membrane to spread out the concentrated stuff, that's osmosis. If you have the concentrated stuff move through that membrane to where there isn't any of the stuff over here, that would be diffusion. But they're both trying to reach equilibrium, get the same stuff on both sides. Facilitating is helping. So facilitated transport is any transport where there are helper molecules. Usually we're talking about proteins that go across a membrane. Tonicity is a fancy word for concentration. So explain how tonicity relates to the direction of water moving across the membrane. Easy. Water always wants to dilute things. Just remember that. Water always wants to dilute things. That means water is always, when it does osmosis, it's going to go from where there is more solute and less water. Sorry, it's going to go from where there's more water to where there's more solute. It'll try to spread that out. So just remember, water wants to dilute things. So you have it concentrated, water wants to go over there and dilute it, spread those solute molecules out. Anyway, compare passive transport and active transport mechanisms. The big thing here is what's passive and what's active. Things that are passive don't require the cell to spend energy to make it happen. You're not pumping things and all that. Active transport, you're spending energy to make it happen. So that's easy. But think about the direction. If you are moving things from an area where it's highly concentrated to where it's less concentrated, you don't usually have to spend any energy. But if you have a bunch of stuff here, you're trying to put more stuff in there, you keep shoving it in, shoving it in, shoving it in, you're going to require more energy to do it. So we tend to use active transport, the transport that requires energy, when we are moving things from a less concentrated area to a much more concentrated area. I tend to compare it to the Japanese subways, where they pay people to shove other people onto the train. They're moving them from the station where there's less people to the inside of the subway where there's more people, and they shove them in. Okay, so you got the idea. At the other end, the subway, you don't have to tell people to get off the subway. They just get off. If they're all packed in really tight, you open the door, they all want to spread out. They move out. Well, molecules are the same thing. Ions are the same thing. They all have the same charge. If you pack them in really close, they'd like to spread out just to get away from each other. Kind of like take a bunch of magnets, put all the negative ends together. They don't all want to hang out. They'll push each other away. So if you create a way for the solute to spread out, they will. Anyway, and then we're going to talk about moving things across the membrane, big things. That's going to be endocytosis and exocytosis. Endo, when we bring things in, and there'll be three types of that we'll talk about. That'll be the penocytosis, or peno means to drink. Phago means to eat. So much smaller items that are dissolved in the fluid when we do peno. And then phago, to eat. That's big things like a white blood cell taking in a bacteria to eat. And then we have receptor-mediated endocytosis. So those are the three types of endocytosis. Endo meaning that we're bringing things in. Exo meaning that we're moving things out. Anyway, that's a high-level preview. Let's jump into the details. So the plasma membrane is a membrane. It's a phospholipid bilayer. You've already heard us talk about that. You know, heads to the outside, tails to the inside. The tails hate water, the heads love water. Okay. And when you put these phospholipids in the water, they're going to line up in that double layer automatically, and when they do, it keeps the outside water out and the inside water in. That's pretty nice. Okay, so you've got that. There's the pretty picture. All the blue things are the phospholipids. You've seen phospholipids before. What you see here are transmembrane proteins. Trans means across. These are membranes. These are proteins that cross the membrane. You can see that some of them are anchored in place to the cytoskeleton, the network of protein fibers, and some of those are anchored in place. But some of them are not. If you didn't anchor it in place, a transmembrane protein would probably stay where it was anyway, because the top end, let's use a mouse, the end right here, it's water loving, and the end right here, it's also water loving. It's also hydrophilic. This middle is hydrophobic. It doesn't want to be near the water. So that means that if it started to drift out of the membrane, just like water would push it right back into the membrane. If it started to drift out towards the inside of the cell, it would move back. Anyway, that's not true of all transmembrane proteins, but most. Any membrane that crosses any protein that crosses the membrane is a transmembrane protein. This would probably be a channel. Let's see what it says. Others call it an integral protein because it's an integral part of the membrane. Anyway, but yeah, a channel protein tends to be like a tube that things can pass through. Remember, things are going to pass from high concentration to low concentration if you're not spending energy to make them go. All right, so moving back, the plasma membrane is made of all these phospholipids. They say the consistency of an oil at body temperature. That actually depends on how many kinks you have in the fatty acids that make up the tails. But that detail doesn't matter for you guys for this class. We're not talking about weird animals in the deep sea. Anyway, a fluid mosaic model. What that means is that the unanchored proteins can move around in that membrane. And sometimes you'll get a group of them that move together in like a little raft made out of cholesterol. Got a Robert Krulwich video that I should probably throw in for you guys to see. Hold on a sec. Okay, I just went and got that video pulled up so I can put the link down below. It's the Robert Krulwich one. It's a short news clip of him talking to Richard Liu from Harvard and how they use computer animation to make biology more understandable. Anyway, but the video in question is on a cell and parts of the cell, which of course is relevant to what we're doing now. And at the end, you can see that whole raft of proteins that are moving laterally in the membrane, which is what that point was. Anyway, moving on. Glycoproteins, glycolipids, those are, you can see the word protein, so that makes sense. It's got protein in it. Glyco means sugary. What we're talking about are these things here. This is a glycoprotein. It's got a protein part that's in the membrane. You've got a sugary extension that's sticking up, a carbohydrate extension that's attached to the membrane and, attached to that protein sticking out. These are part of the membrane. Now, glycolipids would be the same type of thing where you have a sugary extension sticking up, but it would be attached to a lipid. And of course, a lipid is going to sit in those phospholipids really, really well. It's easy for that to be part of the membrane. All right. These things that are on the surface, they're part of your immune system. Or let's see, these are the things that are recognized by the immune system. These are the things that on your red blood cells would be oh, I have A blood, or oh, I have I have Rh negative. Those are based on glycoproteins that are in the surface of the red blood cell. When we say A, it's just a description of what version of a glycoprotein you have. If you say it's an O, that's, oh, I don't see that one. You don't have the ABO glycoprotein. All right, so let's move on. So the plasma membrane has some transmembrane proteins that act like channels. You have a drawing of one right there. And they will allow some things in but keep other things out. A lot of this is based on the charge and the size. Obviously, if a molecule is too big, it can't fit through a narrow channel. So some things will get through because they're small enough to, and others can't. Some things will get through because they have the right charge, and what I mean by that is, if you know how opposites attract and likes repel, if you have a channel right here and it has a bunch of positive charges around the edge, it's going to make it hard for, even though it's the right size and this positive ion could pass through it, it probably isn't going to go, because as it gets close, it'll be repelled by the charges around the ring. Anyway, so that tends to be how. Small things, hydrophobic things, go right through the phospholipid layer. We tend to call that simple diffusion. It's not facilitated, it's not helped. The small things, nonpolar things, they can just pass right through. Bigger things, they need help. Remember, we said that water doesn't cross the membrane really well because water is polar. Well, sometimes you have channels called aquaporins that let the water through. Anyway, let's move on. Here we have an aquaporin, and you can see that the water can go from the inside to the outside, from the outside to the inside, even though it's charged, even though it's polar, even though it wouldn't normally be able to cross these phospholipids really easily. So small things, non-polar things, they go through really well. Big macromolecule, it's probably not going to get through without help. All right. So let's talk about about movement, about how things cross the membrane. Diffusion, I already told you, is the movement of solutes, little things that are dissolved, ions or molecules. Osmosis is the movement of water, the solvent. It can help you remember. This, to think that O. Osmosis begins with O, and osmosis is the movement of water. Facilitated diffusion is anything like you just saw on the previous slide. Facilitated diffusion is diffusion that needs facilitation, needs help. Facilitate means help. So this water has trouble crossing the membrane on its own, but there's a helper molecule, this aquaporin channel. And it makes it easier for the water to move. So that's an example of facilitated diffusion. Well, I shouldn't say that's an example of facilitated osmosis. Okay, but we have the same thing, sodium channels. We pump all the sodium to the outside of a nerve cell, and it stays there until the nerve is ready to fire. When the nerve is stimulated, the sodium comes back into the nerve. But it doesn't come in randomly across the membrane, because sodium is charged. It's not going to be able to cross the phospholipid layer. Sodium comes through a sodium channel, and that sodium channel, being the tube-shaped protein that goes through the membrane, is the helper, the facilitator. So we would consider that to be facilitated diffusion. It's diffusion because the cell is not spending energy to pump the ion. It's diffusion because the ion is going from high concentration to low, but it's facilitated because it has to go through a tube, through a helper protein, which is a channel, a sodium channel. We'll talk later on about their awesome trapdoors. But anyway, Active transport is moving things, using energy to move things. So the sodium potassium pump that put the sodium on the outside in the first place, it's powered by ATP, and it pumps the ions to where we want them. And again, endo is taking things into the cell. I already told you there are 3 types. There's pinocytosis for liquids and very small things dissolved in the liquid. There's phagocytosis for big things like white blood cell eats the bacteria. And then there's receptor mediated, where it's like the picky eater. I'll pull that into the cell if it matches this receptor, if it's something I specifically wanted. Otherwise, not coming in. Exo is a term for the X should remind you of exit. This is cytocell. So exocytosis, things are exiting the cell. And hold on just a second. All right, let's continue. First, we're going to talk about diffusion. That is when molecules are moving, or ions are moving, from a high concentration area to a low concentration area. They're going to try to spread out. A lot of times this happens because they got charges and you don't put all positive things together. They're going to spread out because they repel each other. This will continue until you reach equilibrium. That's when they're equally distributed. The cell is not spending energy on diffusion. But if you hear the word diffusion, you know that this is passive transport. The cell is not spending any energy. The net movement is going to be from high concentration to low. And at equilibrium, you got the same movement into the cell as out of the cell. Great. All right, so here we're showing initial conditions. And in initial conditions on slide 3.8, you can see that we have a lot of stuff on one side of the membrane and not as much on the other. You can see in slide 2, we've reached equilibrium. We've got the same conditions, the same concentration on both sides. That type of movement would be passive. Now, osmosis, you already know the O tells you it is the movement of H2O. And now we're going to talk about tonicity as we do this. When we say tonicity is just a discussion of the concentration. So hypotonic means below the concentration. But what are we talking about? We're comparing the solution outside a cell to the solution inside the cell. So when I say hypotonic, it is, well, they better have a good picture here. Yeah, they do. Okay, that'll work. When I say hypotonic, we've got a cell that we put into a solution that is less concentrated. Let's say it's almost pure water. And when that happens, remember I said water wants to dilute things? Water will cross the membrane, osmosis, and the cell will swell up, all right? That's what you're seeing here, hypotonic. And it is possible in some cases to swell the cell up until the cell pops. Animal cells especially. Bacterial cells, that's harder because they have a cell wall. We haven't talked about that, but they have a cell wall. Plant cells, they have a cell wall. Animal cells, no cell wall. So you can inflate them. If you put them in a hypotonic solution, they can puff up until they burst, until they lyse. Anyway, so that's hypotonic. And if the cell pops, it lyses, it breaks. Hypertonic is the opposite. You take the cell and you put it in a solution that is hypertonic. That's a solution that is more concentrated than the inside of the cell. And again, water wants to dilute stuff, so water will come out of the cell into the hypertonic solution, trying to dilute it. The cell will run out of water, it will shrivel, it will shrivel, it will shrivel, it will shrivel. And So cell shrinking, we call that crenation. There's a picture of it here. You might wonder why it gets all weird and spiky when it shrivels. It's because that plasma membrane is being stretched over the cytoskeleton, this protein fibers that are all over inside the cell. And as the water flows out, that membrane gets stretched over them, kind of like if you've ever seen around Valentine's Day, they'll put roses in balloons. Imagine if they let all the air out and the rubber, as the balloon shrinks, the rubber gets stretched over the thorns. You can probably guess crenation can kill cells. That is actually, before we had refrigerators, is to preserve meat by putting it in salt. And that was the idea, was to have the fluid come out of it, the water come out of the meat into the salt, and then it's hard for bacteria to live there because it's kind of dried out. Anyway, moving on. They're not saying it here, but iso means same. So isotonic right here, this is the cell just being happy. Animal cells really like isotonic solutions. Plant cells like slightly hypotonic, but animal cells, like your blood cells, they like isotonic. They like the solution the same inside and out. And when that happens, you might have a little movement of water out of the cell, but if you do, you're going to have movement that's equal to it in. It's going to be nice. It doesn't overinflate the cell like a hypotonic. It doesn't shrivel it up like a hypertonic. All right, yeah, we've got that. So osmotic pressure is what drives osmosis. Osmotic pressure is a term that we use for water wanting to dilute things and moving from where it's less concentrated, moving from where the solutes are less concentrated to where the solutes are more concentrated as it tries to spread them out. This is important when we talk about things like your capillaries. When you get to the end of the capillary, at the front of the capillary, water leaves the capillary very easily because of pressure from the heart. We call that hydrostatic pressure. But at the end of the capillary, water's flowing back into the blood, into the capillary, because all the proteins and things were and cells were left inside the capillary. And so the blood at the front of the capillary is kind of dilute. As you get further down the capillary, it's getting more and more concentrated until eventually the osmotic pressure makes the water come back into the capillary because water wants to dilute stuff. All right, moving on. Facilitated transport is transport that has facilitation, that has help. So molecules being helped across. It says here, transport from higher concentration to lower. In other words, this is still passive. This still doesn't require the cell to spend any energy to move things. Nothing's being pumped. And as you know, it says here, the transporters are very specific, only move certain molecules. That's based on the shape and size and charge of the hole that things are moving through. Now here, this one is a glucose transporter. And that is a really famous example because that one it actually changes shape. It's not pumping. What's happening, if you think about it, the glucose and whatever else that is, the red thing, I don't know, they come in and they try to touch it and go across from high concentration. Sorry, I'll use the mouse. From high concentration to low concentration. They want to go there. Well, the glucose can, because it's the right shape, to fit inside the transporter. And the transporter, because of the way the glucose is shaped and the charges on it, it is attracted to that glucose. It goes like that. But when it does, that opens up the rest of the passage to get in. And the glucose moves through, and then there's no glucose, so it snaps back to its original shape. What I'm saying is the cell is not pumping this across. The cell is not spending energy. The glucose coming in, when it hits, it causes a shape change to go across, and then things go back to the way they were. The best example I can think of for this is at an amusement park where you go through that turnstile, the thing with the 3 prongs, and as you go through it, it counts how many guests there are, it twists. That is not pumping people in through the gate. People going through it are powering it. The turnstile's people counter is powered by the passage of people. Same way. The glucose transporter is powered by the glucose coming through it, but it helps the glucose get through. So it's a facilitator. It's facilitated transport because it helps the glucose get through, but the cell is not having to spend energy to move the things through, so it is not, so it is passive transport because the cell's not spending any energy. So passive transport, facilitated transport, facilitated diffusion. Diffusion because the things are going from the high concentration to the low concentration. All right, let's move on. Active transport. Now, I'm going to make a little fib here. They point out here, they say, it uses ATP as energy. That's, in honesty, that's a most of the time thing. That's not an all the time thing. Like active transport of hydrogen ions in mitochondria, it's not powered by ATP. In fact, it's used to make ATP. But we'll talk about that later. But they're right. This is most of the time. Most active transport is powered by ATP. So the A in active can remind you of the A in ATP. The A in active transport should remind you that we are spending energy, usually ATP, to get the things to happen. And so there is again, a helper protein, but this helper protein is a pump. We put energy into it and it pumps. And that's moving things from a less concentrated area to a more concentrated area. Like it's already got a bunch of stuff and they've all got charges and they're repelling, they're trying to move out. And you say, I don't care, you're taking even more, we'll put more stuff in there. And you spend energy to do it. So here's an example. It's a sodium potassium pump. This is a famous one. This is the one that's in your nerve that puts the sodium on the outside and the potassium on the inside. You can see we already have more sodium on the outside than we do the inside. We already have more potassium on the inside than we do on the outside. But we're going to keep moving it, and we're going to do it by spending energy. All right. So if we're moving large amounts of things, we're either going to do endocytosis or exocytosis. Endo if we're bringing things into the cell, exo if we're moving things out. So endocytosis, you got 3 types. You got phagocytosis, which means cell eats. Cyto is cell, phago is eat. So phagocytosis, cell eats. It takes in a big thing. And that example that they're using, white blood cell taking in a bacteria. Pinocytosis is cell drink. So it might help you remember if you're a wine person, like a wine snob, a Pinot Grigio, Pinot Noir. Okay, Pinot means to drink, and cyto still means cell. So pinocytosis is cell's drink. All right, and that's, again, we've said it before, taking in little things. Receptor-mediated endocytosis, that's the picky eater version. That's when the cell takes in things, but not randomly. It takes in only what it was looking for. It has receptor molecules on the surface sticking up, and you have other molecules on the outside of this cell that come on, they bounce, and they touch the receptor, but they're not the right thing. But if the right thing touches, it sticks. And then the cell's like, ah, yes, and it pulls it in. That's receptor-mediated endocytosis. All right, here we got pretty pictures of all this happening. Phagocytosis, we're taking in a big thing. And in the case of white blood cell, then we're going to have a lysosome come and fuse with it and kill the bacteria. All right. Here, pinocytosis, we're pulling in fluid, little things dissolved in the fluid, that's all good. Here, receptor mediated, those little orange things are the receptors. And when the molecule, the specific molecule we're looking for, binds to the receptor, the cell says, ah, yes, that's what I needed, and it pulls it in. All right. Exo is making things exit. The EX should remind you of the word exit. So exocytosis, things exit the cell. Stuff fuses to the plasma membrane and it's spilled out. If you watch that Krolwich video that I have there, you can see a computer animation of exocytosis. When the transport vesicle is carried by the motor protein up to the plasma membrane and fuses, stuff inside is spilled out, exocytosis. All right, here's to check your progress. I'm going to pause for a second, and then we can continue. All right, let's do this. So we're going to talk about the different organelles and their parts, the nucleus and the endomembrane system. When we say the endomembrane system, what we're talking about is the endoplasmic reticulum and the Golgi apparatus and the transport vesicles that take things from the ER to the Golgi and from the Golgi to other places. So it's all of these things that are made out of membranes inside the cell. Now the nucleus, it's got a double layer membrane around it called a nuclear envelope. So you've got a membrane, you got another membrane. That whole thing is around the nucleus. That's called a nuclear envelope. All right, anyway, let's move on. So summarize the function of the organelles. We'll go through those. Obviously, mitochondria is super famous, powerhouse of the cell. Little ATP factory, actually. Explain the role and the location of ribosomes. Well, we have free ribosomes and we have bound ribosomes, and then we have the weird ones that are off in organelles that are slightly different. But ribosomes are protein makers. I like to compare them to little chefs that make protein. Oh, that reminds me, I should throw up the RNAi video for you guys again, in case if you didn't watch it before. Hold on. Okay, let's move on. So the nucleus has the DNA, and that's the instructions for making the proteins and carrying out all the things that need to happen inside the cell. The endomembrane system, remember the word endo means inside. So the endomembrane system is the stuff that is inside, and it's made out of membranes. And basically, we're going to be talking a lot here about the rough ER, the smooth ER, and the Golgi apparatus, and the vacuoles that can move things around. All right. So the nucleus has chromatin inside, DNA wrapped around histone proteins. Little sequences of DNA that have instructions how to do something are called genes, and those are usually instructions to make a protein. Just like all the stuff inside of just under the plasma membrane was cytoplasm, the stuff under the double-layered nuclear envelope is the nucleoplasm. The nucleolus is the dark spot inside the nucleus where we are reading the DNA and using the instructions to make RNA for ribosomes. Ribosomes are part RNA, part protein. All right, moving on. So here we have the whole thing drawn. We have our nuclear envelope. We have our rough ER covered with those yellow ribosomes. We have our smooth ER without ribosomes. And Golgi apparatus should have been in there. I've always seen that included in the endomembrane system. But understand that, look at this nuclear envelope. Well, you see it has some of its own ribosomes. I want you to also see that the outer layer, the outer of that double membrane, when it folds and changes direction, we now call it the rough ER. These things are contiguous. They are together. They're the same membrane. It just has different names in different parts. So we've got that. And you know, rough ER covered with ribosomes, that's going to be a place where we make proteins. You know, smooth ER, no ribosomes. So what are you making there? A lot of things you make are lipid components for the membrane parts of the cell. You also tend to detoxify alcohol using a smooth ER. All right, so the nuclear envelope is a double membrane. It's got holes in it called nuclear pores. And those nuclear pores let things go in and out. If you watch that Krolwich video that's right there, sorry, the Nova RNAi video that's down there, in their analogy, they think of the nucleus as the castle, and the nuclear pores are the windows in the castle. All right, moving on. So ribosomes, I already told you, are part RNA, part protein. It's a special RNA. It's an RNA used to make ribosomes, so we call it rRNA. rRNA for one subunit, protein for the other subunit. The bigger one tends to be rRNA, the little subunit on top, the protein. So since that Nova video talks about the ribosome as the chef that makes, follows the recipe and makes the protein, I tend to think of it as the main part of the ribosome is the rRNA. But to be a chef, you also have to put on the poofy hat. That's the protein, the little cap of protein. All right, so there are free or bound ribosomes. And in reality, they can go back and forth. So a free-floating one can join up to a rough ER, stick to it, make the protein it needs to, stick it inside of the rough ER, and then detach and float away. Or if it's something that doesn't need to be protected from the cytoplasm, it can just read the messenger RNA, and make the protein. Anyway, so ribosomes are where we make proteins. They could be bound to the rough ER, or they could be floating around. If a whole bunch of them are together, polyribosomes. That looks like almost like a string of pearls. You have a string of messenger RNA that's being read, and you have a ribosome, followed by a ribosome, followed by a ribosome, followed by a ribosome. Whole mess of them going down there. So it looks like the string with the pearls, the different ribosomes. And like I said, the ribosomes that are attached to the ER are making proteins that need to be shielded from the cytoplasm. So they're going to a different place. They're probably going to the Golgi apparatus to be processed. All right, let's move on. There we go. Remember I said, okay, so this is just the rough ER. This is not the full endomembrane system. That's why they didn't show the Golgi apparatus. All right. So we've got Golgi apparatus. That's a packaging and distribution center for the cell. You make things in the ER and the endoplasmic reticulum, and you send them to the Golgi apparatus. A lot of textbooks draw them as like they're floating from the ER to the Golgi in these little vesicles. That is so not true. Watch the Krolwich video, the ABC News video, and you will see that transport vesicle It's attached to a motor protein. That motor protein is walking down a microtubule, carrying behind it a transport vesicle full of stuff. There are roads, I'm talking about the microtubule. There are trucks, I'm talking about semis, I'm talking about the motor protein that walks on there. So yeah, I should have said there are, a better way to say it is, just like a truck, a semi drives on a road, a motor protein moves down a microtubule. And just like that truck will have a trailer behind it full of stuff, that motor protein will have a transport vesicle that it's hauling that's full of stuff. Anyway, let's talk about this. So the rough ER is rough because it's covered with the ribosomes, and the ribosomes are what make the proteins. The smooth ER makes lipids and chemicals that detoxify alcohols. The Golgi apparatus, they're flat, multiple sacs. Each one is a separate chamber with separate chemistry, and they help the protein fold, change into its final shape. Because if a protein doesn't have the right shape, it doesn't work. It could be even worse than not having it. It could be damaging. So you already know the Golgi Packaging and Distribution Center. All right, here we have the full endomembrane system, and that's much better. You can even see the we're having endocytosis. We're taking in a thing that we want to digest. We bring in, it's trapped in a little vesicle, and all of a sudden a lysosome comes along, fuses with it, and spills out its digestive enzymes, its acids. This big thing gets broken down into little pieces that we can use. Which, let's face it, we take in a big meal and we have all the enzymes and the acids, and we break it down into little pieces that we can use. It's the same thing. What you're seeing right here, like you said, oh, they show it like it's floating. Oh, that transport vesicle is actually being walked by a motor protein. This transport vesicle is being walked by a motor protein. It would probably be good for you to know that things come into the Golgi on the cis face. The C and come in, and the C and cis, that should remind you that things arrive on the cis face. And then things leave on the trans face. Why? Because they're being transmitted across the cell to somewhere else. Actually, because trans means across, and you've now gone across the Golgi apparatus. But however you remember it, cis and trans, good to know. All right, so vesicles are little sacs of membrane. They're used to move things around. Lysosomes, I already told you, like a mobile stomach unit. They're full of hydrolytic enzymes. They're full of acid. They will fuse with a transport vesicle and destroy whatever is inside of it. Fuse with an endocytotic vesicle and destroy what's inside of it. They digest it, break it down. So a good example here would be white blood cells, like macrophages. It's a type of white blood cell that the name literally means the big eater. Okay, so moving on. Oh, here's another check your progress. I'm going to pause this for a moment, and you can check to see you can answer this. If not, roll it back, rewatch this section of the video, and then we can continue. All right, let's move on. So right now we're going to talk about cytoskeleton, moving things around in the cell. That makes sense. They're probably going to talk about the motor proteins that walk on the microtubules. And cell junctions, how we move things from one cell to another. So again, we're going to talk about the microfilaments like actin, the intermediate fibers, and the microtubules, which are the biggest. I know they have micro in the name, but everything in a cell is small. Trust me. The microtubules being tube-shaped, they are the biggest. And those are the ones that the motor proteins walk along. All right. So we'll talk about those. Talk about flagella and cilia. Those are for moving things around. Flagella, think like the tail on a sperm. Cilia, think the little hairs that are on the cells lining your windpipe, your trachea, to move mucus that has bacteria and things up away from your lungs. Yeah, that would be a great example. Or in the fallopian tube, the cilia creating a little current going trying to move the egg from the ovary towards the uterus. Anyway, they'll go over tight junctions, gap junctions, how cells stick together, how we anchor them together. Let's advance this. All right, so the cytoskeleton is full of protein fiber. It is made out of protein fibers. They hold the shape of the cell, they anchor it in place, and they move things around. There are three kinds. The biggest one is the microtubule, which is big enough to be tube-shaped. The intermediate filaments are the ones in the middle in size, and the thin ones are, I've always seen them called microfilaments, but actin filaments, that's fine because they're made out of actin. Actin filaments are the same protein. You might hear that when your muscles get shorter and thicker, what's happening is you have actin filaments and myosin filaments sliding past each other. Same protein, actin. Really thin, two little dimers, so microfilament. So you got two strands and no more. Anyway, let's move on. Microtubules are made at the centrosome. That's controlled by the centrosome. They help with the shape of the cell because they're big and they're thick and they're strong. It says act as tracks that organelles move along. You already know that it's actually motor proteins moving along them, hauling the organelles. It doesn't have to be a transport vesicle like I was talking about. If you need to move a mitochondria from here to there, it doesn't just drift over. You walk it to where it needs to go. Motor protein walking down a microtubule, this time hauling a mitochondria behind it. All right. And during cell division, these centrosome, the centrosome has two centrioles, and they move apart to the poles of the cell and then grow spindle fibers that, actually I shouldn't say that, they move apart and grow spindle fibers at the same time, so it's more of like that. But anyway, you get these microtubules of the spindle that attach to the chromosome, and like at anaphase, where the chromosome tears apart, it tears apart because those microtubules are shortening. Anyway, let's move along. So actin filaments, microfilaments, very long, very thin, and they're involved in movement. Probably talking about like an amoeba, where you're pushing, where you have parts of the cytoskeleton pushing on the plasma membrane. Anyway, the intermediate filaments are in between in size. Intermediate tells you they're in between in size. So expect to be able to put those in order from smallest to largest. Number 2, intermediate, is going to be the easy one. All right, so cilia and flagella are both to move things around. They're both made out of microtubules. Cilia, there's the example that I used before. You're moving mucus that has bacteria in it up to the back of the throat, away from the lungs, because you don't want a lung infection. And you can then swallow that down to your stomach, let the acid fry them up, kill the bacteria. And flagella, sperm, again, same example. All right, there we have the pretty pictures. And 9+2 is how most science nerds describe the structure here on the flagella. You have 9 pairs around in the middle, 9 pairs around a pair in the middle. Anyway, So the extracellular matrix, this means stuff outside the cell. It's not just the plasma membrane and then you're done. Outside the plasma membrane, you've got proteins, cytoskeleton-y type things, fibers go along, and sugars and things like that. They surround the cell that is making them. And in some cases, like collagen, that's and elastin, those are things that make, let's say, elastic cartilage. Those are things that are important there, or your skin's ability to spring back after it's been pulled. That very important thing is based on things made by the cells and pushed out into the space around them. Fibronectin, adhesive protein, sticks to the integral protein. So you have the membrane, you have the integrin in the membrane, and then you have the fibronectin that sticks to that and holds the thing in place. And yeah, self-signaling. So here you can see we have our phospholipid bilayer, we have our transmembrane proteins. There's a lot more stuff going on though. We have cytoskeleton on the inside that are attached to those membrane proteins. We have cytoskeleton on the outside. Oh, I guess we shouldn't call it cytoskeleton. We have extracellular matrix on the outside that has those proteins attached to it. All right, moving on. Now, there are three main types of junctions between cells. We have adhesion junctions where the cells stick to each other. We have tight junctions that form a barrier. A good example of that would be in your stomach. You don't want that stomach acid getting down between the cells and digesting the cells laterally, because the top of the cell that's next to the lumen, the space inside, it's got a basic mucus on it to help protect it, but the sides don't. So we stick them all together super tight to each other, and try to keep the acid from getting down between them, because if the acid got down between them, it would destroy. It would kill the cells. In fact, that's one of the ways we get ulcers. There's a bacteria called Helicobacter pylori that is sort of corkscrew shaped, spirochete, and it will down between cells, and it will leave an opening that acid can get in, and then cells die off laterally because they're not defended. So you get an ulcer. Anyway, tight junctions stop that kind of horrible thing from happening. Gap junction is a spot where it says two channels fuse. Let me say it easier. We got this cell, we got this cell, and they are stuck together because their channel proteins are going across both membranes. It's like having a chemical, what happens in this cell goes into what happens in this cell. If you build up chemicals here, they'll pass through into the other cell. A great example of that would be the intercalated discs in heart cells. If this one contracts, this one's eventually going to contract because they're chemically connected. They have tubes running through from one cell to the other. All right, there are the pretty pictures. You got it. Adhesion junctions just hold them together. Tight junctions is like you riveted the two cells together. And gap junctions, they're like you have a mesh or a colander. There are a lot of holes that go from this cell into that cell. Plants are full of that. Not that that cares, because it's not that you care, because it's a human bio class. All right, there is a check your progress 3.5, and I'm going to pause for a sec while you check yourself and play with the dog. Oh, he says I don't want to be on camera. Play with the dog, and then we will continue. Oscar, you're embarrassing me. All right, let's move on. Okay, energy reactions. We are going to be talking here about metabolism and energy reactions. So let's just jump in. I'm a little baffled because I'm like, oh my God, this could be a huge thing. All right, so we'll talk about enzymes. I want you to know that enzymes speed up reactions by lowering the activation energy, lowering the amount of energy it takes to run the reaction. And summarize the roles of aerobic and anaerobic pathways for energy generation. If you can do anaerobic, if you can use oxygen, you can get a heck of a lot more energy out of, say, a sugar molecule you're breaking down. Anyway, let's do this. Let's jump in. All right, so a metabolic pathway, this is when you have all these chemical reactions, and this one goes, and the thing made by enzyme A is then used by enzyme B to make something else that we call 2. So basically, the thing made by A is used by B, the thing made by B is used by C, the thing made by C is used by D. One reactant, so A makes 1, and 1 is used by B to make 2, and 2 is used by C to make 3, and 3 is used by D to make 4. This would be a metabolic pathway, and the product of one reaction of one enzyme is used as the reactant for the next reaction. This tends to be how we get things done in the body, I mean, for real. If you use a lot of enzymes and work stepwise, you can get things done that normally you wouldn't be able to do. And if you want to make that go faster, you put all those enzymes close together in the membrane. All right, so an enzyme is a protein. It's a protein that speeds up a reaction by lowering the amount of energy it takes to go. So you speed up the reaction by lowering its activation energy. If you see ACE in the end of an enzyme's name, in the end of a chemical's name, it's probably an enzyme. So enzymes, the things they work on are called substrates. And in this case, they're talking about lipase breaking down lipids, or lactase breaks down lactose, things like that. Active sites are places where enzymes grab the substrate. So you have the main body of the protein, and you have one spot that's super important that grabs onto that substrate, that thing is going to work on. That spot that has to be the perfect shape to grab the substrate is called the active site. And it's what makes it specific. Like, I can grab this, but I can't grab that. It's because of the shape in the active site. Enzymes aren't used up in a reaction. They're not a product. They're not a reactant. They're a little machine that makes the reaction go. It makes it go faster. So we can use the enzyme again and again and again and again. Here are some pretty pictures. And let's see. In this one, we're doing degradation, and this one, we're doing synthesis. All right, so synthesis, understand you could have an enzyme, and it takes two things, and it brings them together, and you get a product. What's going on there is those two things, they could stick to each other and form the product on their own. But it's really unlikely because they probably come in at the wrong speed and bounce each other off, or in the wrong orientation, and one bounces off the other. An enzyme takes this substrate, and it brings them together at the right speed in the right orientation so they'll stick to each other. That's synthesis. Degradation, you're doing the opposite. You take this thing and you break it apart. A lot of times that'll happen because, say, this enzyme grabs this, it grabs this, the enzyme changes shape slightly now. We call that induced fit. And when it does, it strains the bond. It makes it more likely the two pieces will pop apart. That's why it takes less energy. Or in the synthesis example, this induced fit then goes, shoves the two together, make it more likely for them to bond. Okay, so enzymes lower the activation energy. They lower the activation energy is just the energy that you need to get the reaction to go, to start it. Think of it as, hey, your gasoline is full of energy that could be used to drive a car, to power a car. But it doesn't work if you don't get the spark in there to start the release of energy. The spark is your activation energy. All right. Some enzymes have helper molecules that are not made out of protein. Coenzymes like vitamins or cofactors like minerals are both helpers to the enzyme. Like the enzyme's close to the right shape, but it's not perfect, so it can't grab the thing in its active site. But with this vitamin present, now you have the perfect shape. So the enzyme plus the vitamin is the active enzyme that can grab onto the thing it needs to work on. Now, we told you an enzyme lowers the activation energy. What we're showing you here is the blue line is how much energy it takes to start a reaction. And this orangish line is how much energy it takes to start a reaction with an enzyme. And you can see the enzyme means that you have a lot less energy needed for the reaction to go. That's why the reaction runs faster. All right, so now let's talk about mitochondria. I know. Mitochondria, powerhouse of the cell. It's totally true. But the way I like to think about it, the mitochondria is like a power plant, okay? I was here when we had 3 hurricanes plow through Polk County, and in one summer. And when that happened, we were out of power for a while. And, you know, there were people who, like Anheuser-Busch shut down their bottling plant and they brought water. They bottled water and brought us water to drink. And Publix brought us ice so the food in our refrigerators wouldn't rot because, you know, the everything was shut down. Well, during that time, if someone had given me a lump of coal has energy in it, but it's not going to do me much good. I mean, maybe I could burn it in my grill and cook with it, but my air conditioning, it runs on electricity. My lights, they run on electricity. My water heater, it runs on electricity. My house runs on electricity. So if you give me some other form of fuel, some other form of energy, and I don't have electricity, I can't really use it. Your cell is the same way. Your cell runs on ATP, adenosine triphosphate. And you may have glucose in the body. Yeah, glucose is an energy source, but it's not what most of the cell runs on. It runs on ATP. So where am I going with this? Easy. That coal, if someone gave me a lump of coal and I can't use it, the power plant down the road, we have a coal-fired power plant over on I can't remember the name of the Lake Parker. We have a coal-fired power plant over on Lake Parker. It burns the coal, it burns the coal. In fact, let's be more specific. It takes that coal and chemically breaks it down by combining it with oxygen. It burns it. And in the process releases energy that is used to make electricity, the form of energy that I can use to run my house. The mitochondria is the power plant for the cell. It helps with the breakdown of sugar by combining it with oxygen. That's that close to saying burning the sugar. But it combines it uses oxygen to break down the sugar further. And as it does that, it makes ATP, adenosine triphosphate. And the ATP is what powers the active transporters, what powers almost everything in the cell. This process is called cellular respiration. It's going to use oxygen and give off carbon dioxide. So mitochondria need oxygen. Mitochondria give off carbon dioxide. There are actually 3 steps in the breakdown of a sugar. There's glycolysis that happens outside of mitochondria. And then in the mitochondria, we have the Krebs cycle and the electron transport chain. Both of those need oxygen, but they need it for different reasons. The mitochondria has 2 membranes, an inner one and an outer one. You already know that. We already talked about endosymbiosis, how it's like the outer membrane may have been supplied by the host that took them in. Anyway, they got a double membrane. The inner one is folded into cristae, and those little cristae, those folds, it gives you more surface area, so you can pack it full of enzymes to do reactions. And yeah, endosymbiosis. Most scientists think that mitochondria were prokaryotes. You already know they have their own bacteria like DNA, their own bacteria like ribosome. You already know they can reproduce on their own inside the cell. Yeah. All right, and there they are. Yay, pretty picture. So let's talk about the ATP-ADP cycle. ATP is adenosine triphosphate, and ADP is adenosine diphosphate. Basically, we have ADP, adenosine diphosphate. It's got 2 phosphates on the end. And if we can phosphorylate it, that's fancy saying we can attach another phosphate, we suddenly make ATP, adenosine triphosphate. And that takes energy, but whenever this unstable molecule falls apart, you have ADP left and a free phosphate that goes off to do other things. When we get energy from food, we can phosphorylate to make ATP. Everybody good with that? All right. So there's a pretty picture. It says the same thing. Cellular respiration, we got 3 parts. There's glycolysis that happens outside the mitochondria. It does not need oxygen. Now we have the 2 steps that happen in the mitochondria, the Krebs cycle and the electron transport chain. They both are in the mitochondria. They both need oxygen for totally different reasons, but they both need oxygen, and they both happen in the mitochondria. So know that either of those reactions in the mitochondria needs the oxygen. They release the energy slowly because you go through many steps. Hold on. Okay, I just pulled up a video that I can put in the, the links below, but it's about using sugar to power model rockets. All right, that to me is the best example where people say, oh, if you released all the energy in glucose at once, there'd be a tremendous amount of heat. Yeah, you cook the cell, you destroy the cell, because how much heat is there? Enough to launch a rocket, okay? So by going little by little, little by little, little by little, we can release some energy and use it. Release some energy and use it, instead of releasing it all at once and destroy the cell. So enzymes, that's their great thing they can do. They can let us get through gradually. All right. So glycolysis takes a 6-carbon glucose and breaks into 2 3-carbon pyruvates. All right. And this happens outside the mitochondria. This happens in cells that don't have mitochondria. And like I said before, it doesn't need oxygen. It produces NADH and 2 ATPs. Nice. Small yield. This is enough to make a bacteria happy to run it. Your brain's a lot more metabolically intense. Your brain needs a lot more energy. To make you, to make me, to make us work, we need to take in oxygen. Why? Because we need to power the mitochondria so they can get all the energy possible out of that sugar. out of that glucose that we're going to break down. All right, so here we have a cell that is bringing in glucose transporter. It does glycolysis, that's taking it from a 6-carbon glucose to 3-carbon pyruvates. And then we are going to move the, we're going to take the remains of the pyruvate and we're going to put them through, we actually are going to modify the pyruvate. We'll go from being 3 carbons to being 2 carbons. Acetyl-CoA is what we're going to make. And acetyl groups are 2 carbons. So I don't know how much detail there wants to get in here, but if you need to know, every time we break a bond, we harvest the electrons to use later to make that we're going to use in the electron transport chain. So that initial break, you harvest some electrons to use. The NADH that they were talking about right there, it carries those high energy electrons. Okay. Then when we go to acetyl-CoA, we turn the pyruvate to acetyl-CoA. Well, guess what? We just broke carbon again. Broke a bond between 2 carbons, and that released high energy electrons that we then harvest and move off to use in the electron transport chain. Now, the citric acid cycle, the Krebs cycle, there's a lot of breaking bonds between carbons, harvesting the resulting electrons to carry off and use in the electron transport chain. You might wonder what's so magical in the electron transport chain. You see the, oh my God, look how many ATP we're making. Because here it's like O2, O2, and oh my word, it's a 32 to 34. What's the difference? We're taking those high energy electrons and using them to pump hydrogens. And then once we have this concentration of hydrogens, they spin a molecule called adenosine, ATP synthase. Thank you. They spin a molecule called ATP synthase, and what it does is it takes an ADP and a phosphate and it slams them together to make an ATP. So that's how the electron transport chain pumps hydrogens. The hydrogens power the ATP synthase. The ATP synthase makes a buttload of ATP. Moving on. That's a technical term, by the way. All right. So when we have oxygen, the pyruvate is going to be modified into acetyl groups that we use for acetyl-CoA. We keep reusing the coenzyme A as it carries acetyl groups. If we don't have oxygen, we will do fermentation. And that's the thing that, yay, it gives you beer and all, but that's also the thing that, oh, ow, we get lactic acid and my muscles hurt. Why? Because as you were running, you were spending more energy than you could make with oxygen, so you had to do it the other way. And this other way, not real efficient, and it generates painful lactic acid, but at least you got to run away from the bear and escape. Moving on. All right, so the citric acid cycle, the Krebs cycle, this happens inside the matrix of the mitochondria, and it says completes the breakdown of glucose by breaking bonds between carbons. Remember, every time we break a bond between carbon, we want to harvest those high energy electrons that are released. We want to reuse those. We're going to use them in the electron transport chain. So we harvest electrons to use later. So each pyruvate is going to go into the Krebs cycle as an acetyl group, a 2-carbon, that we attach to coenzyme A. And when we do this, that's the first time we generate carbon dioxide. Basically, we break the bond, harvest the electrons. And what do we do with this carbon that we got rid of? We combine it with an oxygen and breathe it out. That's how we get rid of the carbons. All we're doing is breaking the bond to take the electrons and then breathing out that carbon as carbon dioxide. All right, and that's the reason why Krebs is needing oxygen, is just to turn those carbons into carbon dioxides that he can breathe out to get rid of. So the electrons and hydrogens are carried away by NADH and FADH2. Those are carrier molecules. They are carrying the electrons and the hydrogen. This is an amazing molecule if you think about it. It has the high energy electrons that are going to be used to pump hydrogen, and the hydrogens that you're going to pump, all together in the same molecule. It carries that off to the electron transport chain to use. All right. Says fats and proteins can be converted into compounds that can enter the citric acid cycle. Oh, let me say that the easy way. Your brain runs on sugar and nothing but sugar most of the time. But a lot of things in your body, even though sugar is the main thing, sugar is what we're talking about here, the breakdown of glucose, you can break down other substances and turn them into things that are partway through the cycle and get some of the energy out of them. So fats, energy storage, all true. We break down the fats and use them to make energy-rich molecules so we can finish and go through the electron transport chain and pump hydrogens just like normal, which power ATP synthase just like normal, which gives us ATP, and then yay, we're still alive. All right, so electron transport chain is using the NADH, and FADH2, even though it's not on here, but those compounds being made by glycolysis, being made by Krebs, or citric acid cycle, they're carrying electrons and hydrogens off to the electron transport chain. And the electron transport chain is made out of proteins that are part of the membrane in the cristae of the mitochondria. And it says each carrier accepts 2 electrons, passes on to the next carrier. Yes, but they're not mentioning the important part. And pumps a hydrogen, okay? It pumps hydrogen across that membrane. You're storing hydrogen in the intermembrane space, and that hydrogen wants to go back through to the matrix. Oxygen is the final electron receptor at the end of the chain. What we're saying there is we've used up all of the energy in these electrons. Well, who would still want them? Well, oxygen is super electronegative. That means it wants to get electrons. It wants to be negatively charged. It will take these old used up electrons. Oxygen is very electronegative, so it's very hungry for these electrons. It takes them in. And then you have the, and to do that, we take the O2 we breathe in, we break it apart, and they take an electron. But if you break those apart, then you have nasty, scary free radical oxygens around too. So we combine those with hydrogens to reduce the number of hydrogens on this side. That's super important because we're letting hydrogen go from the high concentration to the low concentration. And when the hydrogen was down at the in that low area, if it reached equilibrium, we'd stop making ATP. So by neutralizing free radicals with hydrogens, we make harmless water, and we keep the flow going. Anyway, the electron transfer chain releases a lot of ATP, the majority of ATP. So we could get 36 to 38 ATP if we do the whole cycle start to finish. if we don't have oxygen, we can't do Krebs. We can't do electron transport chain. We can't even turn the pyruvate into the acetyl or the acetyl-CoA. So we're still going to do glucose. We're still going to get some ATP, but we're not going to get nearly as much ATP, like a net gain of 2 ATP per glucose instead of 38. That's not good. It's not enough to run you. You can do a little bit of that as a helper to the main system, though. And that's what we're doing whenever we run in and we generate lactic acid. All right, so there's a check your progress. I'll let you go over it, and then I'll continue in a minute. All right, let's move on. Oh, and that's the end. Okay, we are actually done with the cell and membrane transport and energy reactions. So I will leave you now.